Hello and welcome. This is Columbia Basin Research's second video in a multi-part series on using Program SampleSize. SampleSize is a program created by Columbia Basin Research at the University of Washington School of Aquatic and Fishery Sciences with funding provided by the Bonneville Power Administration. SampleSize allows users to input a range of release sizes or estimates to calculate the precision of survival projections and view those results via a graphical interface. Today, I'm going to walk you through the terminology used in Program SampleSize. Let's start by firing up the program. Alright, so if you watched our last video, then you've already installed Program SampleSize. So let's start that up. Head to your search bar and type SampleSize and run the program. Maximize the window. There you go. There's some terminology that you'll need to be familiar with when you're using the program, and those words include detection, tags, mark recapture, site, and reach. Detection is used interchangeably with the word recapture. We are talking about the process of determining whether a specific animal is present at a site. This can be determined without actually recapturing the animal. Tag is used in place of animal, fish, species of interest. A tag is a unique identifier for a specific animal, thus can be a unique marking, a pit, radio, acoustic tag, etc. Mark recapture is a type of study where animals are tagged, marked, released, and then recaptured at a later period or periods. Sample size is designed for these types of studies. A site is the physical location of a release or detection event. This type of study design could also take place through time, i.e. a comparison of two release groups, one in spring and the next 2 weeks later, both the same location. Site then stands for time. Reach is the distance between two sites. Release to detection, detection to detection, etc. As noted above, time period could be substituted for reach. Now that we've gone over some of the basic terminology, we should move into how we'll interpret some of the variables that are used in this program. To do that, the easiest way would be to call up a single release and look at the variables that we have as inputs here. You can see R0 and R1 here. R is the release size at a given site. R0 is always the initial site. If you go down to S, this is survival probability. This is the probability of survival from site to site. The last reached survival probability is always represented as final S times P in this program. P here is the capture probability. It's the probability of detection at a given site. And D here is the proportion of detected removed from a given site. Here you can see the number of reaches involved is 2, and the number of replicates. Replicates default as one replicate study, but I'll talk about the usefulness of further replicates in future videos. Now let's look at the 4 study designs implemented in Program SampleSize. I have up here a single release. A single release entails a single release site and a minimum of 2 detection sites used to estimate survival from one release to another. If I press this button here, paired release, I call up a paired release study design. Paired release involves 2 release sites or 2 releases separated by time using the same 2 or more detection sites after release. This design is used to estimate survival between the 2 release sites and to reduce any bias in survival due to the release process, i.e. handling mortality. Next, I will call up virtual paired release. A virtual paired release is similar to the paired release study, with the initial release subsequently comprised of tags detected at a detection site, creating a virtual release at that site. A paired release is conducted below the area of interest to obtain unbiased survival to the first detection site, also the lowest release site in the design, after the initial release. This design is used to obtain a more nuanced survival estimate in a smaller area, such as getting dam survival starting in the forebay and ending in the tail race, using fish released far enough above the dam to ensure that the arrival distribution at the dam is typical of the untagged population. Finally, I have a Ricker 2 release. A Ricker 2 release is a model similar to the paired release study design, with only one common detection site for the 2 release groups. This has been video number 2 in Columbia Basin Research's How to Use Program SampleSize, basic terminology used within the program, and an overview of study designs. The next videos in the series will provide direct examples on running single releases, paired releases, virtual paired releases, and Ricker 2 releases, as well as some hints and tips to make the most of your designs. I hope that this video helps you get the most out of Program SampleSize. If you have any questions or comments in regard to this program SampleSize, contact us at web@cbr.washington.edu. Best of luck with your study designs, and thank you.